All right, so now we're going to look at this coin toss lab. If we were here in person and I were here with you, we would actually be tossing a coin in this lab. Because I'm not here, I'm going to give you some data instead. So let's go ahead and read the background together. In genetics, we use Punnett squares to predict the offspring or kids that are possible between two parents. These are called expected results, and they are predictions that are calculated using mathematics or Punnett squares. For example, if the two parents have a baby, we expect there is a 50% chance that they will have a baby boy and a 50% chance that they will have a baby girl. However, I'm sure you know plenty of families that have all girls or all boys. Observed results are those results that actually appear in nature. For example, if two parents have 3 children with brown hair, those would be observed results because they actually happen. Expected and observed results may not always be the same, but they're usually close. So we need to label each thing below as either expected or observed results. So it says here, if a statement's a prediction, it's likely expected. Like it didn't actually happen, but we're predicting it. That is expected. If it actually happened, it's observed. So in A, we have a brown rabbit and a white rabbit have 3 baby rabbits. 2 are brown and 1 is white. Did this actually happen? We can say yes. So because it did happen, we're going to say observed. A scientist calculates that when he flips a coin, he should get 50% heads and 50% tails. Did this actually happen, or is it a prediction? This is a prediction, so this is going to be expected. A researcher flips a coin 10 times. He gets 6 heads and 4 tails. Did that actually happen? Yeah, it did, so observed. And a researcher uses a Punnett square and predicts that the offspring of 2 parents will all have blue eyes. Did that happen? It didn't. That is a prediction. That's going to be expected. All right, part 2. So we're going to use a Punnett square to figure out what results we can expect from 2 parents that are big H little h. You might be wondering, what's big H little h? Well, I'm going to tell you. The big H gene, capital H, is the dominant gene, and it creates the normal version of a protein called hemoglobin. Hemoglobin is a protein and it carries oxygen in red blood cells. The little h gene is the recessive gene and creates the mutated version of the hemoglobin protein. So the protein's not being made correctly. So if an individual has 2 little h's, then we have misshapen red blood cells and a disease called cystic fibrosis. If an individual is big H little h, they will be normal since they have enough of the normal protein in their red blood cells. If they're big H, they're going to be normal since they have 2 copies of the normal gene. So we have to fill these in below. Remember, we said the big H gene is dominant and creates the normal version. So if we have 2 big H's, it's going to create the normal hemoglobin gene. Big H little h. Notice it says here they will be normal since they have enough of the normal protein. So they're making some of the normal protein and some of the mutated protein, but they still have enough normal to be considered normal. If they have 2 little h's, then they are going to have this disease where their blood cells are not the right shape, and that disease is called sickle cell anemia. It's because they don't have this right hemoglobin protein. So I gave you 2 imaginary parents. We're going to see what we can predict from these parents. So we've got to fill in this Punnett square. Filling in this Punnett square. We got to see what we expect. So how many big H do I have in this Punnett square? I just have 1, and that's out of 4 boxes. So 1 out of 4, or 25%. How many big H little h? I have 1, 2 out of 4. So 2 out of 4, or 50%. How many little h? I have 1 out of 4, or 25%. So these are my expected results. Now, what we're going to do is we're going to get some observed results. In class, what we were going to do is use some coins here to represent the observed results. Okay, we're going to flip them and get results. So I have some examples here. We're not actually going to flip coins. But the first thing it says here is column A at the table at the bottom of the page has the expected genotypes from your Punnett square. Check to see if they match. So look at column A here. It says for 4 kids, we would expect 1 big H, 2 big H little h, and 1 little h little h. Is that what we got? 1, 2, 1. Let's go back up. 1, 2, 1. Yeah, that's what we got. Okay, great. So now let's take a look at what the rest of this says. When we do this lab in class, we use coins to represent genes. We use 2 coins. So notice 1, 2. 1 to represent the gene in the egg and 1 to represent the gene in the sperm. Remember, Punnett squares are showing us what are the possible combinations of sperm and egg. Each side of the coins, there's 2 sides, a head and a tail, represents the gene possibilities from each parent. So because each parent was big H, little h, they could either give a big H, which we're going to use as heads, or a little h. So it says heads represents the big H gene, giving the big H gene to the sperm or the egg. Tails represents the little h. So imagine we take these two coins, we flip them up, and we see, oh, we get heads and tails. That's representing, for this possibility, that we have a kid that's going to be big H, little h. We flip them again. Then we get 2 heads, big H, big H. We flip them again. We see we get little h, little h. And then we flip them again, we get big H, big H. So we're going to record these in our table below. So it says the toss results are shown in column C. Look at, I got 2 big H, big H. If I go back up to my tosses, 2 big H, big H. I got 1 big H, little h, and 1 little h, little h. Notice, they don't match up with my expected. That's okay. So these are what I actually got. This is what we expect from our Punnett square. We're going to calculate percent, okay? You probably know how to do this. Percent is you take the number divided by the total. Here there's only 4, so I would have 1 divided by 4, which we know is 25%. Here I would have 2 divided by 4. That's going to be 50%. Here I have 1 divided by 4, 25%. It gets a little trickier over here. Notice my total here is 200. So again, you're going to have to do these as well. But here you'll have 49 divided by 200 times 100. And I have a cheat sheet up here. So you're going to calculate the percent. So remember, your percent should all add up to 100. Okay? Now, then you're going to fill in the claim, evidence, reasoning. So our claim is that collecting more data allows us to get our observed results closer to expected. What do we mean by more data? We mean when we have 200 tosses, it's closer to what we would expect than when we just flip the coin 4 times. Okay, so using your data, when we looked at the data for what, which data in the table, not just the data, which data? And then what did we see? Okay. And then because we saw this, this means that collecting more data allows us to come closer, okay? So again, you're probably going to have to talk about, well, when we had 200 tosses, we got these percents. When we had 4 tosses, we had these percents. And so the 200 tosses were closer. So therefore, that means collecting more data allows us to come closer. Now, you're going to answer these questions. All the answers to these questions can be found in the words, in all of the wording in this lab. So for example, one of them says, why do we use 2 coins? Well, here's your answer. We use 2 coins, 1 to represent the gene in the egg, 1 to represent the gene in the sperm. So if you get stuck, you need to go back and look, okay? And then there's just a few more questions, and that will be it. For this last one here, it says, let's say you do a Punnett square and you collect the data, your observed results don't match the Punnett square results, they're way off. What should you do? Do your best guess here. We'll talk about it when I get back, okay?